welcoming back all the enthusiastic and brilliant listeners of the Immunology for Everyone podcast by SKA. After a peek into nature's role contributing to the very intricate immune responses, let us dive further today into the two pillars of our defense system. SKA is here again giving you introspections into how the entire cascade of immune response proceeds to the reactions our body has towards different pathogens. We dearly value your responses, queries, and interest. Keep listening to these podcasts and do share them with those who will benefit the most out of these. Over to your host, SKA.
Thank you very much. Hello, everyone, and welcome back to our podcast, Immunology for Everyone. I have another very interesting aspect of immunology to discuss today, and that is regarding two important cell types, the dendritic cells and macrophages. We have heard about these cells in previous episodes because I talked about them during innate immune response as well as the antigen-specific immune response during T cell activation. These two types of cells, besides being sentinel cells of innate system, also play the important role as antigen-presenting cells. Both of these cells have the capability to capture the antigen, display on their surface bound to MHC molecules in the manner that it can be seen by the antigen-specific T cells, leading to activation of adaptive immune responses. So this process implies that the generation of an activated T cell response requires the presentation of antigenic peptides on a bone marrow-derived and professional antigen-presenting cell. And also, I would like to respond to a query raised by one of my immunologist colleagues in response to my previous podcast regarding role being played by macrophages if the dendritic cells become maturation defective in a disease condition. And I think it will be important to discuss the overlapping and distinct roles of macrophages and dendritic cells in response to invading pathogens. So let me once again refer back to the findings from our lab, which I discussed a few episodes earlier. And some others also have given that kind of information, which I discussed in one of the previous podcasts in context of chronic viral hepatitis C infection. And there I indicated that a maturation defect in dendritic cells leads to non-availability of activated CD8 positive cells in the liver, which can be a possible cause of chronicity in hepatitis C infection. But couldn't we ask a few questions here? That is, if the dendritic cells are defective in a disease condition like chronic hep C infection, could the macrophages activate the naive T cells? And also, under what conditions or settings would the macrophages initiate the immune responses? And then, are there different antigen-presenting cell requirements for priming of CD4 versus CD8 T cells? Because the dendritic cells and macrophages as antigen-presenting cells may make different mediators and thereby influence responses in different ways. The initiating antigen-presenting cell may play the role in determining whether protective immunity is generated or not. So then we can also ask, are the T cells primed by macrophages functionally different from the T cells which are activated by the dendritic cells? So it will be very interesting to understand the roles and synergies between these two antigen-presenting cells. So it has been described earlier that the T cells require antigen presentation via an antigen presenting cell of hematopoietic origin. And the cells which come from non-hematopoietic origin, they are found to be unable to prime the naive T cell responses, which may be because of the lack of necessary co-stimulatory molecules on them, or they may be unable to traffic to the lymphoid organs. And you remember that the dendritic cell, once capture the antigen, it actually migrate to the nearest lymph node where it interacts with the T cells to activate the T cell. So let us try to understand what are some specialized functions of these two types of cells other than just antigen presentation. And in this context, we now know very well that dendritic cells are the most potent bone marrow derived antigen presenting cells, and that they reside in most of the tissues in the body. On the other hand, macrophages are also another bone marrow derived antigen presenting cells that can present the antigen and also express the co-stimulatory molecules. And macrophages are also found in most of the tissues, and their numbers are rapidly, increased at the sites of infection or inflammation. So let us see unique features of each one of these cells. If we talk about macrophages first, so besides antigen presentation, these cells also play a very important role in inflammatory processes because they secrete many pro-inflammatory cytokines in response to a pathogen or a damaged tissue. So in addition to their critical role in antigen presentation, they are also maintaining the homeostasis by uniquely acting as sentinel cells, which are responsible for clearing the cellular debris, and they also promote the tissue repair, which is a very important function, which is very unique of macrophages. And we also know that many tissue resident macrophages are present in various organs of our body. For example, the alveolar macrophages are found in the lungs, microglia in the central nervous system, Kupffer cells in liver, red pulp macrophages in spleen, and so forth. They are performing tissue-specific functions, but they are essentially, the phagocytic cells and a type of macrophage. So these tissue-resident macrophages, they persist through the adult life as a self-maintaining population, and they are involved in homeostatic and organ-specific functions. So in order to replenish the pool, the hematopoietic stem cells, they basically act as the major source of monocytes, which are the precursor cells for macrophages, and which not only replenish the tissue-resident macrophage population, but also get recruited following injury and infection. Then another very important characteristic of macrophages is that these cells display remarkable plasticity, and they can adopt a spectrum of phenotypes by responding to the signals in their surrounding environment. Like, for example, during an inflammatory response, large number of circulating monocytes are recruited to the site of tissue injury in response to various cytokines which are released, like interferon gamma. And these cells, they differentiate into a inflammatory, classically activated macrophage. And these inflammatory macrophage types are called M1 macrophages. So M1 macrophages, they produce a lot of antimicrobial products like nitric oxide, reactive oxygen species, interleukin-1, tumor necrosis factor alpha. So all these, molecules, they have antimicrobial effect. They also express very high levels of MSC class 1 and class 2 on their surface, and that way they become very good antigen presenting cell. However, when the inflammatory stimulus is eliminated, there are other factors which are released by the immune cells, such as pro-resolving lipid mediators or Th2 type of cytokines, which are include the IL-4, IL-10, and all. So these factors, they influence the macrophages to become more of anti-inflammatory phenotype. And that anti-inflammatory phenotype is called the M2 macrophage. The M2 macrophages are mainly reparative macrophages because they promote fibrosis and help in resolution of inflammatory responses by producing many factors like matrix metalloproteinases, growth factors, and the cytokines like transforming growth factor beta. So these all help to resolve the inflammation and do the tissue repair. So in addition to all these factors, they also express arginase 1, mannose receptor, PDL2, and the immunosuppressive cytokine, which is IL-10. So these all facilitate the restoration of homeostasis. So you can actually appreciate the polarization of M1 to M2 and then back, even ex vivo. You can take the monocytes, convert them into macrophage, and then culture them with bacterial lipopolysaccharides and add a little bit of interferon gamma. You will see that all the macrophages will become M1 type. They will start producing inflammatory cytokines. Or if you give interleukin 4 and interleukin 13 in the tissue culture, and all the macrophages will become M2 type. So M1 and M2 macrophage phenotypes, they can interchange very quickly depending on the cues in the environment, and that's how they respond. Initially, it was thought that there are two different, binary cell states of these macrophages, but recent data has described that macrophages in vivo can express markers associated with both phenotypes, and they can immediately convert to either M1 type or M2 type as and when the environment changes. And so these macrophages can also be termed as either pro-inflammatory macrophages, which are the M1 type, or pro-resolving macrophages, which are the M2 type. The complexity of macrophage activation within the context of a disease will emphasize their unique ability to respond to dynamic changes in the environmental stimuli. So let us come to now dendritic cells. Dendritic cells are basically relatively recently described. It was Ralph Steinman who first described dendritic cells in only 1970s, when he found these cells in the spleen. But later on, it has been discovered that these cells are present in all lymphoid as well as most of non-lymphoid tissues as well. So prior to this discovery of dendritic cells by Ralph Steinman, immunologists generally thought that the macrophages were the main antigen presenting cells in the immune system. This may be because compared to dendritic cells, macrophages are more abundant in numbers. They are evenly distributed throughout the body in various tissues, while dendritic cells are rare in number. They are only 0.1 to 0.5% of the total leukocyte population. So it took almost 10 years for people to accept that dendritic cells are the professional antigen presenting cells. So these dendritic cells are named because of their probing tree-like dendritic shape. And these cells are responsible for initiating the adaptive immune response. And so they are also called the sentinels of immune system. Uh, these are bone marrow derived leukocytes, and they are most potent as antigen presenting cells. Although dendritic cells are also very heterogeneous population, because but some of them, they are specialized in capture of the antigen, processing of the antigen, and then converting the proteins into smaller peptide molecules which are loaded onto the MHC molecule. And then these cells, they carry the antigen and become mature, and they present it to the T cells. So in addition to immunogenic roles, I discussed earlier also that these are the cells which play a very crucial role in maintaining the immune tolerance to self-antigens, because they are the cells which present the self-antigens to the autoreactive T cells. But at that time, they do not express the co-stimulatory molecules. So the single signal through MHC molecule, peptide, and T cell receptor leads to generation of energetic T cells, and these T cells become tolerant to the self antigens, and that maintains the central tolerance against autoantigens. So based on their transcriptional programming and functional characteristics, so these antigen presenting cells can be classified into either monocyte-derived dendritic cells, because the monocytes in circulation upon stimulation, they can be involved in the inflammation as well as infection. So these are the cells which are called monocyte-derived dendritic cells, and they can be created ex vivo also. From the monocytes, you can isolate from the blood, and then you can develop them into monocyte-derived dendritic cells. Then there is a population of conventional dendritic cells, and these are the cells which recognize extracellular as well as intracellular pathogens and present the peptides to CD4 and CD8 positive cells. And then we have plasma cytoid dendritic cells. And these cells, they normally exist as plasma cell kind of morphology, but once activated, they change their morphology to dendritic cell type, and they are very important for antiviral responses because they produce tons of type 1 interferon. So in fact, they produce almost 99% of type 1 interferon under those conditions when the pathogens are invading. So these cells, they differ from macrophages morphologically as well as phenotypically. And since dendritic cells have numerous cytoplasmic processes, they have a high surface area permitting intimate contact with large number of cells which are surrounding them, like T cells or NK cells or neutrophils or epithelial cells, because they have large surface area. And experimentally, it has been shown that one single mature dendritic cell can interact with from 100 to maybe 3,000 T cells at one point of time. Whereas macrophages are not like that. So the dendritic cells and macrophages are two different types. That's what I'm trying to describe. So these dendritic cells, they normally exist as immature cells, and in all the non-lymphoid tissues, they are continuously looking at the pathogens, and once they look at it, they capture it, and they further mature as they migrate to the draining lymph nodes in response to the chemokines, I told you. And those are the chemokines like CXCL19 and CXCL21, because these cells, as they mature, they start expressing chemokine receptor CCR7, and they also produce inflammatory cytokines like TNF alpha and IL-12. and they are in the subcapsular sinus of lymph nodes. These dendritic cells, they interact with the naive T cells and present the antigen for activation. So these properties of dendritic cells, the immunoregulatory capacities of dendritic cells, they hold a great promise for the treatment of cancer, autoimmune diseases, and prevention of transplant rejection. And these have been actually exploited. Like the manipulation of dendritic cells could turn them into most effective adjuvant of to enhance the host immune response. We take the example of cancers because tumors have been shown to suppress the dendritic cell activity by secreting anti-inflammatory cytokines like IL-10. So the dendritic cells which are present in the tumors, they become suppressive type kind of dendritic cells. They are not able to perform their function of presenting the antigen and activating T cells. So in order to subvert these kind of mechanisms, the dendritic cells could be generated ex vivo, loaded with the tumor antigens, and then re-injected to boost the host immunity against the tumor cells. So these are called the dendritic cell-based vaccines, which can be generated in this way. And these are quite safe and have very minimal side effects and have proven to be feasible and effective in some patients. There are other strategies of exploiting dendritic cells in various disorders, and these have been described, and there are many clinical trials are being investigated to find these cells. So now let us address another very important question, and that is, are the dendritic cells only cells which can prime the naive T cells, or macrophages can also do the function? Because the general understanding now is that dendritic cells are the cells which can stimulate primary T cell responses and may lack the be particularly the potent cells in doing so. Whereas the macrophages, they lack this capability, but the literature is quite limited about it. It is possible that the dendritic cells are the principal antigen presenting cells for presenting antigens for some pathogens, because most of the pathogens, dendritic cells play a professional role of antigen presenting cell. But in contrast, it has been demonstrated experimentally in some of the reports that macrophages can also stimulate the naive CD8 positive T cells in vivo. Now, whether macrophages by themselves are also sufficient to prime CD4 cells, the literature is quite scanty. There are very less understanding about this question. So is it likely that macrophages can stimulate primary T cell responses to only certain antigens? That is also a very important question to be addressed, which has yet not been addressed very clearly. So although there is some information available, yet it is shown that in vivo, the macrophages can acquire some antigens either directly from the lymph nodes and spleen or in tissues, and then migrate to lymph nodes and activate the CD8 cells. So there are some more such reports present in the literature. For example, if we immunize the animal with macrophages which have engulfed some pathogenic antigen, then we find that these cells can prime naive T cell responses also. But in these experiments, it has not been well described, because it is known that dendritic cells they can acquire the cells which have been given as immunization, and they can cross present the antigen. So it is not clear whether these are the macrophages which are directly presenting the or priming the T cell responses, or it is the cross presentation by the dendritic cells. However, there are some studies where they have used toxic liposomes to deplete all the phagocytes from these animals, which indicates that macrophages also play an important role in initiation of naive T cell responses, but under only some circumstances. So the information available so far indicates that presentation of antigen by macrophages to T cells is important for the effector phase of immune responses. That is when T-dependent activation of macrophage is there. Because whenever there is a macrophage T cell activation, the T cell also releases cytokines like interferon gamma, which activate the macrophage. And in these conditions, the activated macrophage really sends out activating signals for the T cell, rather than in the priming phase uh, of the responses, which is stimulating the naive T cells. At that stage, the naive T cell is not able to really send out the activation signal to the macrophages. So the question still remains. Now, but it has been shown that under some circumstances, macrophages can also activate naive CD8 positive T cells. Now the further question comes in, are the macrophages and dendritic cells the only cells that can initiate T responses, or there are other cells also? So in this context, it has been found that even fibroblasts, which when they were transfected with viral proteins and injected directly into the spleen, they were also able to generate viral specific cytotoxic T lymphocytes. So these findings, they would suggest that any antigen bearing cell that migrates to lymph nodes can initiate the T cell responses. But since many experiments have demonstrated that bone marrow derived antigen presenting cells are necessary for initiation of immune responses, so fibroblasts are unlikely to play a role in priming of the cell responses in physiological situations. However, in the artificial experimental conditions, they have shown that they can prime the cytotoxic T lymphocytes. Similarly, people have done experiments using T cells as antigen presenting cells, and that they demonstrate that the ability of antigen bearing cell to migrate into lymph node is not sufficient property to initiate the responses, because T cells as antigen presenting cells do not express all the co-stimulatory molecules, which is also another requirement for activation of naive T cells. So the B cells have been described as antigen presenting cells. They can prime T cell responses in vivo, although this has also been quite controversial at the moment. So it is an interesting and important finding that besides dendritic cells, macrophages can also initiate immune responses under some circumstances. So these observations raise many important questions. And I reiterate, in what settings do the macrophages undertake priming of naive T cells? That is one question. And then, are the T cell primed by macrophages functionally different from T cells primed by the dendritic cells? And finally, it will be also very interesting to examine the interactions and synergies between different antigen presenting cells. And before I close, I encourage the budding immunologist among listeners of these podcasts to take up such areas of research to resolve some of such queries which are there, and we keep asking questions. So with a hope to bring in some more interesting questions maybe next time. Till then, goodbye and thank you very much.